Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요. And welcome and thank you, SynBYSS people, for joining us. The 124th seminar, and I call it a UC Berkeley Day. As I announced yesterday, after more than a decade of experience of Washington University leadership's discrimination and harassment and retaliation against my students and me based on rationality, gender, religion, and race, which have been intensified after the racial discrimination incident committed by the Washington University Engineering Dean and BME chair, as well as the cover-up efforts by other leadership people, I was forced to quit my Washington University position. The university has been preventing me from using my $6 million grant money, talking to my lab members and colleagues, preventing hiring new members, preventing submitting proposals, and even accessing to the campus for more than 2 years just to make me quiet and cover up their crime or helping cover up their crimes. They also sabotaged my new job hunting by spreading lies and framing me as a crazy and dangerous person. They even sent armed policemen who almost killed me in my office. Unfortunately, my 3 years of efforts to change the toxic culture failed, but I will continue to make big efforts to achieve this goal. I'll do so in a new institute, J. Craig Venter Institute. The JCVI, in short, my new home, cares about diversity, equity, and inclusion, as well as, of course, is a world-class research institute led by Dr. Venter, who has huge scientific contribution to the society, including the first human genome sequencing project and the construction of the first synthetic cell, as they claim. I'm super excited to leave the toxic environment and join this excellent and collaborative institute, although I've never imagined leaving my first job. Okay, it is my tremendous honor to introduce very briefly, but probably that's impossible, our speaker, Professor Wenjun Zhang. She does not need any introduction because we all know her. She is a professor at UC Berkeley and a world expert in de novo biosynthesis of bioorthogonal functionality. I think she and I met for the first time in person, but before I knew her, I think that's in 2016 when both of us were invited to attend and give a talk in China. More importantly, or coincidentally, one of her former students now, Antonio Del Rio Flores, he's amazing, by the way, now at Stanford University as a postdoc, who will be joining CU Boulder, I think the Colorado, as an assistant professor, was my official AICHA mentee, and I gave some career advice to help him get a job. My just minimum help and advice, and more importantly, Wenju's many years of mentoring for him led to his securing a faculty job even before he started a postdoc. That means Wenjun is great mentor, and Wenjun got education for future leaders, and I really appreciated her for her effort to mentoring, nurturing these future leaders, and absolutely she's amazing. Wenjun, thanks so much for your dedication and contribution to the education of future leaders and scientific advances. And as I said, I couldn't introduce her short. It's become longer. Your podium is all yours now. And thank you again, Wenjun. It's absolutely wonderful and to have you today.
Well, really amazing to hear you serving as a mentor for Antonio too. Really very appreciated.
Easy. He's great. So I didn't just say whatever you want to do it, and then you will get the job. And then he got the job, multiple offer. I already know because, and then he was considering big university, and then oh my God, he got everything, in one just shot, and that's amazing.
Okay, so I guess I will start by sharing the screen.
Please.
Okay. Is this a full screen you are seeing?
Full screen. Wonderful. Go ahead. Thank you.
Great. So it's really a pleasure to be invited here as a pioneer speaker to start today's session. First, I will start to share some of my research vision. My lab in UC Berkeley has been focused on natural product research for the past decade. Today, I will talk about how this research can inspire the human health and synthetic biology. I will start with the definition of natural products. These are bioactive small molecules produced by diverse natural sources such as plants, microbial organisms, and marine organisms. And collectively, they have played important roles in treating nearly all human health conditions. This is historically why people care about natural product research. But I would say the impacts of the natural product research go well beyond this traditional view. I will first take an example of human microbiome research. So now we know as a human body, we host at least 10 times more of the microbes than our own human cells. Indeed, many of these microbial organisms have so-called accessory genomes, often differing in the specialized metabolism. And these differences can dictate traits of these microbial cells to be pathogenic, commensal, or even probiotic. Here, there are also many potential natural product biosynthetic gene clusters on the genome, as an example shown here, found from the specialized metabolism. And these biosynthetic gene clusters have been found to be correlated with human health or disease. But in general, we know very little about the activity of these gene clusters. For example, what are different structures of the encoded natural products? What are the downstream social chemical relationships these natural products will mediate, and what are their immediate activities? Then I will share one of our recent examples to highlight our research. This is a collaborative research where we looked at an oral probiotic initially isolated from saliva of a kid from New Zealand. And from this oral probiotic, we identified a natural product biosynthetic gene cluster. And then we further reviewed the natural product produced by this gene cluster. We named them salivibactins. We also found salivibactins have potent antibiotic activities towards many human pathogens, including gas, the group A streptococcus, here both in vitro and in vivo assay in these assays. So gas is a well-known pathogen which can cause life-threatening diseases such as flesh-eating disease, or more mild diseases such as strep throat. Therefore, we were very interested in looking at the interplay of this probiotic and gas, which can naturally see each other in our mouth. And by studying the regulation of the biosynthesis of this new antibiotic salivibactin and how they interact together, based on this knowledge, we further engineered this probiotic to overcome the pathogen defenses and to prevent colonization of this pathogen from various in vitro and in vivo models. So today I wanted to highlight this example because later in the longer talk, you will also hear another fantastic engineering effort on microbes for more of a biosensing applications. Well, here, the study of this new antibiotic salivibactin can also further benefit from synthetic biology because although it looks small, it represents a new molecular scaffold, and chemical synthesis is not straightforward. A naturally producer cannot produce a lot for us to further study its mode of action or to develop as a drug lead. So here, synthetic biology can really help us. We can introduce a potential biosynthetic genes into a heterologous workhorse organism such as E. coli in different combinations, where we can further understand the biosynthetic pathway of this molecule. Meanwhile, we can also optimize these biosynthetic pathway to produce a lot more of this molecule for downstream study. Another area I think natural product research can also benefit synthetic biology is to discover useful and unique enzymes to generate different unique functional groups on demand. So here we can also engineer these enzymes to install these functional groups on different molecular scaffolds. For the past decade, my lab has been focused on many unusual bioorthogonal and clickable functional groups. And here I'm showing just one example of terminal alkyne, which we know the alkyne azide-based click chemistry has revolutionized the field of chemical biology. Here, by studying the biosynthesis of this natural product, Jamaica-mite B, which naturally contains this terminal alkyne functional group, we reviewed this portable tri-gene cassette in producing this terminal alkyne functional group tethered on the carrier protein. And then also by using different engineering strategies, we can selectively target the different natural product pathways to directly generate alkyne tagged natural products. Here I'm showing one unique application of this de novo biosynthetic platform too. Here, by utilizing this azidophlorogenic probe, we can realize the in situ quantitative readout of the generated alkyne tagged natural product for the different downstream applications. So this project was initiated many years ago by my former graduate student, Wenjun Zhang, who was bravely enough by that time to join a just established assistant professors group. And now she's also assistant professor in Texas A&M. So this transition to my last topic to talk about my brief advice to young researchers, as I was told to do so. And my advice is mainly focused on the mentorship. I feel this is a key for success for any young researchers, particularly young PIs. Here, I feel, first of all, it's great to work with different diverse trainees, but different trainees, they are all very different. Our graduate students or postdocs might have very different motivations, backgrounds, and personalities. So as a PI or young PI, our mentorship has to be tailored to their need and also to tailor to their different career stages. Example will be the same graduate student comparing to the early stage when the graduate student just joined the group to the later stage, a senior resident in the group. Maybe they will demand a different mentorship style. Again, I cannot emphasize more about communications in the research environment. And also the success looks different for the different trainees. I strongly recommend to use individual development plans to cover various topics in mentorship meetings. And these topics include scientific knowledge, research skills. I guess these two are very obvious, but also to include less obvious topics in regular meetings, such as how to improve communication skills, management and leadership skills, responsible conduct of the research, and career development for young researchers. Lastly, maybe just a quick advice for young PI, how to recruit great researchers, in particular postdocs in the group, because initially maybe it will be challenging. Go to different scientific conferences in your field. I believe these in-person interactions and the relationship is the key to recruit really the best trainees to help you to start your career. I will stop here today. Thank you.
Absolutely beautiful. Thank you. I 100% agree with you about your advice, and then the work just blew my mind, even though that's short talk. And also, I want to invite Caroline Bertuzzi to this seminar series, but once she got the Nobel Prize, I couldn't get. I should have contacted her earlier than she got the Nobel Prize. Anyway, so one thing I want to mention, absolutely, all of that is very important. That's how, Wenjun have many people in academia and as a professor at this moment, I guess, including already I mentioned, two of them. And that's because, of course, their effort, but at the same time, Wenjun's mentorship, that is fantastic, I think. So another advertisement, as you know, we have the December Hawaii conference. So you can apply. We are almost finishing up the website. And then once we finish website, we will release the announcement, and then you could meet a lot of great minds in synthetic biology. Thank you again, Wenjun. Brilliant. I love it.
Thank you.
Thank you so much.
All right. So now, the main speaker of today with a longer introduction. You know what? I tried to make it shorter, and then I just cut it half, but still longer. Dr. Yonatan Chemla. Is it right? The pronunciation?
It's a French CH, so it's Chemla, like Chevrolet.
Chemla. Okay. So Yonatan Chemla complete his undergraduate study in bioengineering at Ben Gurion University in Israel. He then pursued graduate studies there, and he delved into bacterial translation and genetic code expansion using cell-free platforms. Interestingly, I just went to synthetic cell conference that many people working on cell-free platforms. During his studies, he was honored with the Azrieli Fellowship, the premier fellowship for PhD students in Israel. In his thesis, he developed advanced cell-free platform for the synthesis of protein with unnatural amino acid. That's probably some common thing with Wenjun in some sense. Also demonstrated that mRNA secondary structure regulates translation scanning reinitiation in bacterial operons. In 2020, Yonatan was awarded the Human Frontier Science Program Long-Term Fellowship, another very competitive one, for his postdoctoral research, now with Chris Voigt lab at MIT. Of course, he's my also advisor. That means he's my academic brother. Currently, he is exploring how to transition synthetic biology from the lab to the real-world application, And I do believe it is time for synthetic biologists to deliver, because we promised for last 20 years, but this is really the critical time we need to really generate product and then commercialize and then that in the market. Here's a researching method and design principles for domesticating new bacteria pertinent to the field, and also method to genetically modify them. Additionally, he is developing new genetic reporters based on small molecules, which can be remotely detected by hyperspectral cameras to enhance biosensor transduction in open fields. I believe I listened to Chris Voigt's talk about that amazing hyperspectral cameras research, and I'm delighted I will do a longer version of Yonatan's talk today. Again, he's my academic brother, although we do not have any overlap in Voigt lab. Yonatan, thanks so much for your time today. Please take it away. It's all yours now. Thank you.
Thank you. Yeah, thank you so much, Taesuk and Wenjun for these introduction. Let me share my screen. And before I maybe dive in the presentation, can you see my screen, by the way?
Beautiful. Go ahead.
Yeah. Before I dive in, so I won't forget, I just want to comment on Wenjun's amazing work, that we're actually also starting to look at your alkynes, so the natural alkyne products, as an interesting reporters also for the field, not only for the lab, also using hyperspectral imaging. So I just wanted to say that we're connecting there, and your work might be valuable in many different aspects that maybe you thought about it, or maybe you didn't even thought about these avenues. So yeah, thank you for having me, for inviting. And I am fascinated, or I think we all are fascinated by synthetic biology and the ability to engineer living organisms. And we're all here also because in the past 20, 30, even some, if you consider the first insulin product, then you could say 50 years of biotechnology advancement has been incredibly useful and powerful in society. But I would say that there are three main arenas for synthetic biology. The first two, where synthetic biology has actually made a lot of progress, is in the lab for lab demonstrations, right? And we have plenty of examples for that from our lab, for many other labs. And then the second arena is in industrial fermenters under control settings. And this is more of Wenjun Zhang and a lot of other people, where the first synthetic recombinant insulin was produced, when new bacteriocins like saliva, I don't remember Wenjun's the name of the molecule, but where you could produce these natural molecules on scale and put them into the market and improves people's lives and people's health. So in these two arenas, synthetic biology, I think, has already delivered. But in the third arena, and I would argue it's the most complicated, most complex arena, synthetic biology and all the product does not transition well from the lab to the field. And this arena is obviously the field. And when I say the field, I mean it in the widest sense. So it could be anything between agricultural applications and all the way to health related application in medicine, where you want some cells, genetically engineered cells, to behave in a certain way, or bacterial probiotics, or skin microbes, etc., etc. And the reason that I think this gap between lab application and field application is bridgeable, I think you can trace it back to before I started grad school. And here you can see some, before I started undergrad, I should say. So here you can see some pictures I took when taking some gap time and traveling Eastern and Southeastern Asia. and absorbing the amazing wild nature there. So you can see here the famous hills of Guilin in China, or here you can see Angkor Wat in Cambodia. And observing this nature, that was first what made me fall in love with biology and then deciding to study it in my undergrad. But observing this nature, you can see that nature works, right? However, it made it possible, nature finds a way to work and do incredible natural applications in the field. And when you say nature, there is the nature that we can see, the nature that I saw when I traveled Southeast Asia, and that's the trees, the waters, and the animals. But there's also the microbiome, the nature that we cannot see, which is even more complex, abundant, and diverse. And that's what fascinates me, and that's what I want to dive into today. And I think if we know how to engineer this kind of nature to do in a rational, efficacious, and safe way, we can realize great potential. And this potential, this payoff, could take many forms, right? And here I'm just listing a very few possible applications that could be done with synthetic biology. And this is a very small subset. So you can think about agricultural applications like nitrogen fixation, pesticides, plant health, fighting against plant pathogens, right? Bioremediation to heal our environment, biomining, increasing our ability to mine precious metals, precious materials. And we could also tackle applications like self-healing or living materials. We can fight corrosion. And the benefit here is not only the financial benefit I list here on the left, right, which is a very, very big markets that could be potentially disrupted by this new synthetic biology technologies, but it's also, I think, and I would argue, one of the only ways, and maybe the main way, for us to help fix everything that is broken in our planet's health. Everything that needs fixing, I think biology can have a big part in that. So there's an amazing potential, but as I said, there is a gap that is hard to transition between our current understanding and that of the field. And just to give you an example, so this is not new, this realization that I just mentioned. It was actually realized immediately at the onset of biotechnology, of biological engineering. At the early 1980s, already researchers thought about how to make genetic engineering for field applications. And when I first joined the lab, we started kind of diving into this field. So we started working on this very expansive review that we're still working on it 3.5 years later, but hopefully soon it's going to be published. But in this review, we looked backwards in history to see how this technology evolved. And you can see back in the 1980s, there is a lot of interest in the use of genetically engineered organisms or microbes in the field, right? And there was actually very interesting tests and very interesting products that were tried to be approved at that time. But then you see something happening around the turn of the century that basically this interest drop, right? So there is almost a drop to close to zero. And recently, maybe 20 years later, we're starting to see this tick a little bit up. And these are the different applications that were used basically in all these researches, mostly in the 1980s and the 1990s. So the question that we asked ourselves is, okay, these technologies are obviously very appealing for biological engineers. So what happened to basically make it drop? And we found three main reasons for that. The first reason, which is maybe the easiest to fix, but it's also the most inhibitory of all those reasons, and that is the regulation landscape. And that means that basically our regulation in the US, but also in most other places in the world, and we talk about it in the review, are incredibly inhibitory. They were written, most of them, on the 1980s or based on science and understanding of the 1980s. And the leading principle in all of these regulations is the precautionary principle, and they're very risk adverse. That means that it's really hard to get approval to even do an academic field test to, for example, to explore the safety of genetically engineered organisms. So regulation is a big problem. The next one is safety. And that is how we can make sure that these new technologies are actually safe. For example, what happens to the genes we were introducing to the environments, what happens to the bacteria we introduce or the microorganism we introduce to the environment, how do they evolve, how do they affect microbial ecosystems, et cetera, et cetera. These are all kind of open questions. Obviously, there is a lot of good work done in this field, but it's still, we're far away from decent amount of understanding. And finally, the last reason we saw this drop in the 2000s is after all these trials in the 1980s and 1990s, people realized that it's really, really hard to get a genetically engineered microorganism to function and be effective, efficacious in the field. So for the sum of all these reasons, people realized that around the turn of the century, and you can see basically a distancing from these field applications. So what I wanted to do in my postdoc, in my work here at the Voight Lab, is basically look at these three main categories of problems and then start tackling them, or at least understand how to tackle them one by one in a more systematic way. So the first one, regulation. We've been talking and participating in a lot of workshops trying to make regulation, to update regulation to reflect more the data that we have today about the release of genetically engineered organism. The same goes for safety. But today what I want to focus on is my work regarding how to make engineered products or engineered bacteria more efficacious outside well-controlled settings. And to do that, we are developing what we call design principles for environmental applications. And we did a few projects around that, and I'll go quickly through the first two, and then I'll dive in the third application or the third project that we're working on, and then I'll skip the fourth one. And the first one I want to talk about is, or the first question that we wanted to tackle is, do we actually know what happens when we release the genetically engineered bacteria to the environment? Do we have any real world precedents to study to understand what's happening in all the questions that I raised earlier in the last slide? So to do that, we found that the first and maybe the only, there's some nuance when I say the only, but the only release of engineered bacteria to an uncontrolled environment in nature for environmental applications happened in Estonia on 1989. Back then, Estonia was under the Soviet Union. And what happened there was a huge oil mine shell fire here in this Estonia oil shell mine. And this at the time was the largest oil shell mine in the world. And in it, there was a fire that basically underground fire that raged for about 3 months. This entire mine was burnt down. And these are pictures that we took when we visited the mine. This is how it looks like today. And after this was burned, the way that they basically put out the fire is they eventually decided to just flood the mine with water. And by doing that, by flooding the mine with water, they released very large amounts of pollution to the environment through this water that sipped into the waterbed. And mainly the main source of the main concern of pollution was phenolic compound pollutions that were causing very serious health effects in the region. And because of that pollution, the Soviet authorities, together with scientists in the University of Tartu, which is in Estonia, you can see it here in the map, they decided to take a genetically engineered bacterium they developed at around late 1980s. You can see it here. It was developed using a plasmid from a different bacterium. And this plasmid harbors two genes, Phe-B and Phe-A, that could degrade phenol and go all the way from phenol into muconate, and then from there into the central metabolism of the organism. And then you can have a total gain of 9 ATPs. So they had this strain already engineered. And on March 89, they decided to basically produce hundreds of liters of this engineered bacterium and then throw it down into the flooded mine. And they did that. And then they measured the phenol concentration, and then they saw they actually went down. Also, they did some control experiments. So theoretically, it worked to reduce the phenol contamination. And then people kind of forgot about this event. The area got better. There was another field sampling to look for the bacteria. They couldn't find the bacteria. They did find evidence for the genes at 94. And then again, kind of forgot it until September 23, when we went there to Estonia to collaborate with the group that released it and another group from Tallinn University. And basically our idea was to go to the area, sample it, do metagenomic analysis, and look for what happened to the released genes and what happened to the released bacteria, and try to understand it for to study from it for other real world applications. So this is how it looks like. So you can see here, this is the area in Estonia. It's called the Kohtla-Järve region. This is the Baltic Sea. And this, you can see here, this basically big amount of ash. And I didn't say before, but I should, after the fire was put out and all the water sipped away, all the ash from the mine was carried, all the ash and the semi-coke from the mine was carried to this big pile here. And you can see it here in the map. And they built this canal around it to carry all the rainfall, supposedly polluted water, into the canal. And then from the canal, you can see it here, it goes into the Kotla River, and then to the Porsi River, and then all the way to the sea. So theoretically, the bacteria should be here, and they might be also, or the genes might be also here in the canal and in the river, and then all the way to the Baltic Sea. So we sampled the entire area. We took soil and water samples. We then did metagenomic analysis. We did a technique called nested PCR to isolate specific genes and sequence them. And I won't go into all the details, but the bottom line is that we found the genes 35 years later. We found them in multiple species of Pseudomonas. The original release strain was also a Pseudomonas putida. We found it in at least 3 different strains of Pseudomonas. We were able to isolate 2 of them from the environment. And we were also being able to show, if you look here at the map, so you have the orange dots and you have the green dots. The green dots are sites where we couldn't find the genetically engineered or the released genes, and the orange dots are sites where we did find it. And what you see here is actually quite striking. You can see that whenever there is phenol pollution to this day, because this semi-coke mountain is still active, you can see that wherever there is a phenol pollution, you can see the genes because they're being presumably positively selected for. But when you go outside the water system that is polluted, basically these genes disappear. And I think this is a very kind of striking case where you could see that environmental application was both supposedly efficacious, last for long, and also did not actually kind of spread away beyond the environment when you wanted to be active. And then we did some more analysis. So we found these Phe and Phe genes are very rare in nature, and they can basically be found mainly here, almost nowhere else. You can see here this analysis. And finally, we also show that if we retrosynthesize these genes and we put them in Pseudomonas putida, we can get the phenotype of phenol degradation, and they can grow. This is their growth ability, as phenol is the only carbon source. So the conclusion from this is that 35 years on, the genes kind of colonize the area, but only the area where you maybe want them to colonize. And then the second story that we kind of was striking from when we wrote the review I told you about is that it's very common sense, but if you use bacteria not well suited for a certain environment, it will not be efficacious. It will not persist for long, right? For better or for worse. So if you want an application where you want a bacteria to be more efficacious and persist for longer, what you want to do is actually isolate bacteria from that environment and then bring it to the lab, engineer it, put it back in the environment where you want it to function. And this is what we did in this project. We asked the question how we can make bacteria perform better in natural settings through the field to lab and back to the field kind of strategy. So to do that, we went to a different place. This time we went to Israel. We took soil samples from 2 sites, one in the north, one in the center of Israel, and from different depths, different soils. And our idea was to take these soils, characterize them. So we did characterize them, which microorganisms live in these soils. But then our second aim was to engineer them. And the way that we chose to engineer them was using a technology developed here in the Voigt lab called Export. And this technology used Bacillus subtilis, to use its conjugation machinery called ICE, I'm going into all the details, to basically conjugate bacteria directly, or conjugate other bacilli directly and supposedly in the environment. The problem is that it was only shown in lab, right? So in cultures, this technology, and we wanted to see if it's possible to show it for actual field samples. So that's what we did. First thing, we checked the relative abundance of the bacilli in these samples. So these are our engineering targets. And you can see that all samples except T4 had bacilli in them. And then we introduced them to the export donor strain, which basically inject all the soil sample with this genetic payload, which is a very simple payload, very strong promoters, very strong RBSs, and just a GFP and a selection marker. And we try to do it directly on bacteria isolated from the soil and on the soil itself in addition. And this resulted in around 25 different new isolates of different bacilli that were engineered. And then what we did is, instead of spending time in like choosing a chassis and then optimizing it for a specific function, what we did is we basically had a pool of 25 chassis and one genetic design, and then we could just choose the chassis that performs the best out of them. So it's kind of a new engineering paradigm of, instead of optimizing your construct, you just find the host that is performed the best. So we think it's kind of interesting, and we chose this one specifically. Was Bacillus, or its closest reference was Bacillus frigori tolerans. And you can see that it produces a lot of GFP. You can see here from this flow results compared to the wild type. And then we can mix it back with the soil, and you can see that we have a very nice phenotype of GFP expression compared to the native bacteria. And kind of to take it to the next level, we wanted to see if it could persist. How far are these bacteria going to persist in soil, right? And then what is the genetic stability over time? So to do that, we basically put them in these flower pots, we put them in this contained container, into this contained facility of the US Naval Research. They call it a jungle room. It's basically a room that simulates jungle conditions. We put our bacteria there for so it's for about 16 days. You can see it here. So this is how the wild type looks. This is how the engineered bacteria look. And you can see that after 16 days, you can still see a GFP signal. And this is just using a flashlight. This is not like any fancy imaging machinery. But then we kind of forgot about this experiment. We left the pots there in storage in the jungle room. But then we remembered 2 years later that they're still there. And we say, hey, let's check how they look almost 2 years later. And we took them back to the lab, and this is how they look. They're basically completely dried. But when you put them in a gel imager, it's actually quite striking. So you should look on this one. It's actually quite striking. They're still fluorescent. Almost 2 years later, the bacilli that we mixed with the soil is still fluorescent it being in the storage in the jungle room. And then we wanted to see if they're still viable. So we rehydrate them. So you can see it here. So we basically just put some water for a few days, and you can see how the GFP signal basically increases, showing that these bacteria are still viable after all this time. And we isolated these bacteria, and we showed that in terms of their phenotype, right? So you could look at the black cytometer GFP curve versus the green here. And you can see the black is the bacteria that was isolated before putting on the soil. And on the green, it's the bacteria that was isolated after 2 years from the soil, or almost 2 years. And you can see they basically performed the same. And finally, we did the genomic characterization, and we were very surprised to see that we see no mutations in the operon that we inserted. So very little genetic drift, high genetic stability. I should caveat and say it could very well be that it's only under these conditions, and it's hard to generalize these conclusions beyond. That was very interesting. And then finally, the third thing we understood while working on this field, and also specifically working on the last project, is that GFP and other reporters are very inadequate when you try to measure them in the field. They don't work well. And we asked the question how we could read information better from living organisms under field conditions. And when I say read information, I mean it in the widest sense, right? You could think of biosensors, you can think of tracking, you can think of other applications, but it actually goes wider into all information. How do we move information from bacteria to humans in the field? And as I said, current modalities, right? There is basically, if you look at optical reporters, and those are the ones you want to consider, you have three main groups. You have the fluorescence, luminescence, and chromogenic reporters today. And the first two categories really revolutionized biology in the past 30 years, and this is GFP and luciferase. And the reason they revolutionized biology is because of these three items at the end of the list. They have a very high signal-to-noise ratio. They can be measured quantitatively in living cells, and at least GFP does not require to add any synthetic substrates, right? The problem with them in the field, though, is that they use complicated detectors. They're very sensitive to ambient light, which is very common in the field. They're very slow to image, and being slow is not a problem, but it actually limits you from scanning large areas because of the imaging speed and complexity, right? And their energetic behavior is actually quite weak. So they cannot be detected, or it's hard to detect them at large distances. And this goes especially for GFP, less so for luminescence. But luminescence had the added problem that it's actually very energy demanding from the cell. So only very viable cells will produce any good luminescence signal. The opposite of these two is the chromogenic reporters. And these are basically small molecules usually, but it could be proteins that produce color. And these are extremely useful for the field, right? The same way we see color all around us. These work extremely well with very simple, very fast ways to measure them in the field. But the limitation is that they have weak signal to noise. They're not quantitative unless because of light scattering phenomena. I won't go into too much details. And they usually require synthetic substrates to be added, and that is not compatible with the field. So we asked ourselves, can we basically solve these three issues or improve these three issues and making chromogenic reporters be useful for the field? And to do that, we turned to a technology called hyperspectral imaging. And in this technology is basically like your regular cameras, but instead of capturing 3 wavelengths of light, you capture 100 times that. You capture hundreds or 300 wavelengths of light, and that produces images with very high spectral resolution. But basically, for each pixel in the image, you get an entire spectrum of light absorbance. And that lets you detect very slight differences in the signature of the absorbance signature of a molecule. So these sensors are actually increasingly available. It's a new technology that is kind of used for many applications today, and it's accelerating. And you can find a need from anywhere from space to the lab with platforms that you can buy off the shelf today. So we asked ourselves, can we use this technology to basically produce a hyperspectral readout that will be slightly shifted, let's say in this principal component analysis of this image, right? Could we make some pixels here, each dot is a pixel, be different enough that we could detect it by changing the molecular structure of that pigment? And to do that, we needed to know how different small molecules absorb light. And that was actually surprisingly difficult because there is no good database, or there is no good systematic experimental exploration of the different molecules and how they absorb light. So we had to create this prediction pipeline to predict the light absorption spectrum of molecules. That's what we did. We took around tens of thousands of molecules from molecular databases. We extracted the metabolites. We built the 2D. We retrieved the 2D structures. We then predicted the 3D structure. And with that, we predicted the spectrum by using TD-DFT calculations of all these molecules to create this predicted spectral library. And this is actually quite hard. So it's a very computational heavy task to do. So it took us many, many months just to run the computation on these molecules. But we were managed to do it. And here you can see a representative sample of 20,000 molecules and their predicted absorbance spectrum. The x-axis, the wavelength, and the y-axis, the absorption maximum. And you can see that most molecules absorb light in the UV, which is less than 400. And then as you go more towards the red, less and less molecules absorb light. And then we could, using these, we can look for molecules that maybe have a more unique or interesting spectral features, like this example here of bacterial chlorophyll alpha, which we will come back to, that absorbs, basically you can see that it has like 3 different absorption peak, and it has a very red-shifted absorption peak. And finally, the predicted spectra was actually, even in this very complex molecule, the predicted spectra in orange gave us a good indication of the actual experimental spectra that we got when we measured the actual molecule. Okay, so we have the molecules, but that's not enough, right? Because if we want to produce these molecules as reporters, we need to produce them in living cells. To produce them in living cells, we need them to be genetically accessible, or we wanted to assess first, using a given host, using its base metabolome, how many recombinant enzymes do we need to put in that organism, right, to produce the target molecule. And we can run this predictions on all the different metabolites. And then the next step would be to calculate how unique this molecule is, right? So we cannot immediately know from just the spectra if this molecule is unique or not compared to all other molecules. So we did this comparison, and then we can add in another level of how unique it is in comparison to a specific background. And that gives us these 2D plots. This is the output, which the y-axis is the number of steps needed to produce this molecule in a given organism, and the y-axis and the x-axis is how unique the molecule is. And obviously we want this lower right quadrant here, meaning molecules that are unique and are more accessible in the organism. And finally, we use that to build the reporters. Okay, so we have the entire computational framework in place, so we can predict for a given host, beginning with E. coli, what are the best targets to go after? And E. coli was very clear. You could see, so this is the molecule I showed you before. It's really hard to synthesize in E. coli, around 11 enzymes are needed. But this molecule, biliverdin 9 alpha, is easier to produce, only 1 enzyme, and it's predicted to be very unique. So we targeted this molecule, and then the next step is basically to build a genetic circuit to produce it under a sensor, a quorum sensor, quorum sensing sensor in this case. So we build this circuit to produce biliverdin, and you can see that the first objective is complete because we were able to produce it in E. coli, take your images of it in the hyperspectral camera, do some image analysis algorithm that we developed ourselves, and then you can get a very nice quantitative response with chromogenic reporters, with hyperspectral cameras in E. coli pellets. So the first problem is check, we can create genetic reporters. The second one, can we see it over complex backgrounds, or what is the signal to noise ratio? And here also, we basically did some kind of field simulation. We took the sand, we buried a disk with the reporter, with the inducer, with the analyte that we want to sense. And then we spread uninduced bacteria that are producing, so this is Putida in this case, on the sand. And you can see 24 hours later, you get a very nice response of the Putida on the sand using the hyperspectral reporter and the hyperspectral camera. And then finally, we could also do the opposite. So like the project I showed you before, and this will be very quick, so I'll be done in 2 minutes. We could use the same strategy we used before, and instead of putting a reporter in a chassis like E. coli, we could just look for a chassis that already produces a very good reporter. And this is the case with the bacterial chlorophyll that I showed you before. And we found that these non-sulfur purple bacteria, Rubivivax, already naturally produces the reporters. So all we had to do is get these bacteria, find a way to genetically engineer them, and basically put this very unique metabolite under our inducible control in the lab. And we did that, and I won't go into the details of the genetic construct, but we did it, I think, in a very interesting way. And then we can get the production of bacterial chlorophyll as a hyperspectral reporter and this purple bacterium in the lab. And we took this to the field, and we did the same thing as we did in the lab, the different inducer concentration, and we put it in these kind of plastic contained vessels filled with sand, and we put the inducer in these kind of plastic containers, and then we spread the bacteria, we put them in the field. We took a drone at about 120 feet in the air to image these bacteria, and we saw a very nice quantitative response. And the idea is that it also works under field, under complete daylight, so we check mark the other problems that associated with chromogenic reporter that I told you before. And then kind of to push it to the extreme, wanted to see if we could see it from space. So we took this purple bacterium, we collaborated with this army base near Boston, and we built these 4 enclosures that we sprayed 3 of them with the bacteria on different times, and the 3rd one as a control. And we took this actual satellite picture and to see if we can see the signature of this hyperspectral reporter even from space. And a caveat, these are very not ideal conditions. The satellite is not hyperspectral, it's only multispectral. You only have 1 relevant band instead of 100, so the signature is much weaker, and the spatial resolution is really bad. You can see it here, but still, under these limitations, we're still able to detect the hyperspectral molecule even from space. And with that, I will conclude. And just to say that I gave you a few stories of our kind of ongoing quest that will obviously take a very long time, right? I think there's a lot more to learn and to study and to engineer about bacteria in the field, and we need to systematically pursue it. But I think we're making progress, and I think these projects that I show you are small steps in the right directions. And yeah, and with that, I want to thank all of our lab, all my collaborators, especially Itai, which worked with me together on the hyperspectral project, Victoria of the Estonia project, Jessica on the project that I didn't mention, and of course, Chris, which is an amazing mentor that I learned a lot from. Thank you.
Wow, again. So that is again, the last part of the talk is what I heard from Chris. That is absolutely brilliant there. So I have a lot of questions, but let me check whether we have the question from the audience. Okay, let me ask some questions. So as you know, I'm really care about biocontainment. So we working on for many years. So one quick question. For the original phenol degrader story, why do you think they use engineered so-called GMO rather than the natural, bacteria, including my own bacteria, Rhodococcus? They degrade phenol very well without any engineering. But of course, I mean, the Pseudomonas, is easy to engineer, and they also have some ability to degrade phenol or aromatic compound. But I just wonder, because now lots of regulation you mentioned, I also talked to many people, EPA, NIH, USDA, those people, they are concerned about those things. But my question is why? Or why do you think they start using the GMO rather than just native bacteria?
Yeah, that's a very good question. They could theoretically use the progenitor where we got the genes from, right?
Mm-hmm.
They knew about it. They could get it. I think so. I think there's 2 parts. First of all, I will ask them next time I will meet them.
Sure.
One of them is still alive. But what I think is first, their engineered organism were better in the sense that they actually did some directed evolution on them before releasing them to the field. So they moved the plasmid, and then they did the directed evolution rounds to improve the phenol degradation capability. And then thirdly, I think they just wanted something quick because they had it already. They could grow it. They knew how it works. And they wanted to use it. I think interestingly back then, especially maybe in that part of the world, people were less, I guess, afraid of GMOs than they are today.
Yeah, that's more perception or related to some big company doing bad thing. but they
Yes.
also making a lot of GMOs. You know which company I'm talking about. So another thing I want to mention is your data is surprising to me. Oh, before the asking that question. So I know, I mean, the Jen Brophy's work and then the follow work that's about the conjugation. That's a brilliant idea.
Yeah.
Do you think if you manipulate the native bacteria through conjugation in the lab, do you consider that as GMO or that's not GMO?
Huh. But without any recombinant genes, right? Let's say you do
Yeah, so
conjugation
no,
with
the recombinant gene, yes, because you manipulate, let's say, whatever original bacillus that has the engineered gene cluster, you want it to transfer to the native bacteria. So essentially, there are two ways. You could release this bacteria, GMO, that is a donor, but that's a GMO, into the environment. In that case, I mean, there are only one GMO, that's your bacillus, right? If you do so, there will be other GMOs that take up your cargoes. But there are two ways. One is you do, and then you only have one GMO release. You will be blamed by people because you release only one GMO. But if you do so, that conjugation process in the lab, do you think that native bacteria as a GMO? I believe so. But what's your view about that bacteria? That bacteria potentially go back to the native, environment.
Yeah, no, I agree with you. I think it will be considered GMO, but it's a very interesting use case what you propose, and I think people are thinking about it seriously. It will be very hard to approve even for experiment today. Like conjugation system is something there will be there will be approved. But this actually brings me to an interesting follow-up point on that, is that it's actually a little bit of hypocritical, because, and that's interesting, we are releasing copious amount of recombinant DNA and the conjugation systems and everything, right, with industrial waste today. Incredible.
That is my point of the paper. So if you read my papers, it's published in Cell Report Method. My argument, I also, published multiple perspective paper.
Yes, I read most of them.
Yeah, we have been releasing recombinant DNA without chopping up that DNA.
Yep.
Last probably more than 50 years, because we are, it is mandatory to kill bacteria after experiment, but we are not, we are allowed to release, also we release a lot of the DNA into the environment only by denaturing, that is autoclaving, but autoclaving doesn't cleave the DNA at all. That's the
And
data
autoclaving is the best case scenario. A lot of places they just do like pasteurization.
Yeah, also the bleaching doesn't destroy the DNA. So we already released tons of the recombinant DNA that also
Yes.
problematic, because that also, I believe, another small portion of the region, we basically have the antibiotic resistant gene spread.
Yes, definitely.
So that's my argument, and that's why I developed the system, no antibiotic resistant gene containing cloning system, that's published in Cell Report Method recently. But that, yeah, so I mean, still GMO. I mean, even if you have the plasmid that has no antibiotic resistant gene, but still GMO, and then your conjugation method, I believe also people will say the GMO, even though that is done in the field or in the lab. So that's kind of one thing I thinking about probably more than 10 years, but still,
Yeah.
it's a very challenging problem to address.
Yeah, there's many questions, and I'm glad to hear that you agree with that, because people don't think about the fact that there is some double standards, I should say, between how we treat living organisms and how we treat just the recombinant DNA. But it's basically the same thing in terms of horizontal gene transfer. I mean,
Yeah, I discussing with the White House people. I'm discussing with the EPA, USDA and also NIH about that issue for more than 50, I mean that problem is more than 50 years old problem, or even
right.
whenever we start the recombinant DNA work, that we release a lot of DNA into the environment. Okay.
It's a fascinating problem. Also to track it, I really want to try to track it in the environment.
Yes, and then your camera thing, amazing. That's why I'm always kind of, I mean, Chris is visionary in some sense, so I kind of always kind of surprised whenever I want to do something, he already doing something. He's much faster than me in some sense, because you guys is MIT, so you are very productive. So my last question is surprise. I mean, it's surprise to me because, as you know, I done pretty much a lot of literature search, and your first figure showing the GMO in environment, the number of the GMO research drop around 2000. That is true, because that's what I also observed. But I hope to see your paper, because I wanted to do that type of review, but I didn't have time to do so.
Yeah.
So, but I had the extensive literature search before, and then all the conclusion I remember now is GMO with the recombinant DNA do not compete with native bacteria. That is 30 years of the evidence and data. That's what I believe. But you just saying today, based on 35 years later, about that's Estonia, right? You still find no bacteria, but that's engineered, the original GMO, but also variant of GMO. That's my understanding. Am I right?
Oh yeah. No, I should have been clearer. No, we don't find the original bacteria. We find only the genes.
You only see the gene, but that
Yes.
because of horizontal gene transfer, and that they still
Yes.
have. So that's the same idea, because the gene,
Yes.
going around, but that's the consistent, because all the data before, because they focus on the original GMO, the conclusion was no GMO survived. That's conclusion.
Yes.
But the DNA survived, the
Yes.
recombinant, and then this now is consistent, because that was my belief, and that's my data. The 30 years data. Also in my lab, we also doing similar thing what you are doing, and I guess we are also scooped by you soon, because we are not the near end yet. The
Wait,
question I have.
you're doing like long-term horizontal gene transfer
We
experiment?
are testing the kill switch in the real environment.
Amazing. So by the way, your paper, I wanted to tell you, your paper, the kill switch in the gut, in the mice gut,
Yeah.
is the only case that anyone ever tested a kill switch in an actually relevant environment. So I wanted to say that I appreciate that.
Thank you, but I do not consider that's a real environment. We are talking about real environment like Mississippi River sample, soil sample like you guys did. So that is a real environment. That's only the mouse. That's not real environment. That's still in the lab. We are not still
I think
contained.
it's a very good important step forward.
Yeah, of course. So the question I have is, so now I understand that you didn't find any original GMO, but you only find the gene. That's great, because that's consistent with the 30 years of data. However, did you find some potential variant of original GMO. Because especially the phenol case or the original case, the phenol case, that is the gigantic adaptive evolution laboratory environment.
Yes.
Because there are phenol, so phenol basically force bacteria evolve in real world environment. And then that's the only case those GMO will survive because they have the ability to compete with the native bacteria because phenol kill other bacteria and then that's giving selection pressure. So did you
Yes.
find some variant of original GMO?
We couldn't find anything, but so the original GMO, if you remember, was released with these genes on a plasmid.
Mm-hmm.
We did find the entire plasmid, if that's we did find the entire plasmid all over. Yeah.
But that's in the different species, different strains.
Different species. Yeah, we couldn't find. Yeah, we couldn't.
So that's
find
consistent with the previous data set. That's fascinating. How about the bacteria in the whatever jungle?
Yeah.
But that's
about it?
original. What is original GMO, right?
Yeah, but that's actually I wouldn't make too many conclusions out of it because the amount of bacteria we added was
Gigantic.
not gigantic. It was like 10 to the 10 per gram or something insane like that.
Okay.
So I wouldn't. Yeah, and also it was sterilized soil. So
that's that's a different. Okay, so yeah, that's not the competition.
Exactly, yes.
So in that case, I mean, can I say GMO do not survive, but the genes survive?
That is my belief. Obviously, we need more data. We need to generalize it, but that is my intuition, yes. And that is consistent with our data.
Okay, so that's great. So I want to collaborate. So so we are want to do gigantic ecosystem, survey.
JGI, you talking?
No, I want to do the survey, the huge center proposal and grant to investigate the distribution of those kind of GMO genes. That's one thing I want to do. And what you are doing is basically exactly same philosophy or concept. And I have been thinking about that for 10 years, but I focus on more kill switch development. And I already have the now tools in the kill switch and also PNAS paper that is talking about how to engineer. And then now we have some environmental engineers. And at the same time, we have the multiple other people we are working on. So the Chris group with the camera thing, I mean, we can do multiple different things, and then we can solve the problem of the recombinant DNA floating around for 50 years or longer. But by the way,
yeah, I'm sure Chris would love it. I would also love it. Hopefully, if I get my own lab, I would also love it.
Probably that's easier because I intentionally did not collaborate with my own advisor. So I didn't do any collaboration with my own advisor because I want to be independent. But now I do expert, I'm the senior-ish.
Now you're allowed.
Yeah, I'm now allowed. So anyway, I now realize I'm so excited. I'm so excited, but now I realize this is 11:17 the Central Time or 9:17 Pacific Time. So let me check. Yeah, the people go ahead.
Before you end the recording, I wanted to put it on the protocol that I also I think it was in your recent review about this work of environmental. You say something that I think is actually quite genius and visionary, that you could treat like the world, the soil, the water is huge bioreactors. I feel and that this is so simple and it's so smart way to put it. I was really inspired by this statement.
Thank you. So, but that's not new. People think that way. Environmental engineers think that way for many, many years, but nobody put that in the paper because, metabolic engineering field, that is something I would say I don't know the right word for that one. I was not allowed to say so. That's the right word. I was not allowed to say so because in metabolic engineering, we typically think just one bioreactor contain one bacterium, etc. But now I'm senior. I can say anything I want to say, right? So that was my say. So I wait for 10 years to say so. That's my that's what I want to say. I wait 10 years to say so, and then I finally done that. I'm so glad I did that. Okay, so let me close. Thank you all for joining and staying today. We'll meet again on May 9, Thursday, the same time, the same Zoom link. We'll have Professor Ming Hammond at University of Utah and Kei Jing at MIT, another MIT. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I stop recording. Let me see, stop recording.